Hello everyone. Today I will give my presentation about my previous work. It is about the roles of cell fusion between mesenchymal, stromal, and stem cells in malignant cell in tumor growth and metastasis. This is actually my honor project when I was an undergraduate. So I'm going to give a really brief introduction about the cell fusion, which is the background about the whole project. It actually has the potential to enhance the malignancy and also give the normal cell a stem cell-like properties. And basically what we are trying to study is about EMT process. We are trying to make sure the cell fusion will initiate the EMT process and give the cancer cells some worse properties. So cell fusion is actually really basic phenomenon in every kind of species. While fertilization, everyone is from there. But today we're going to talk about the macrophage fusion. Macrophage is actually a really important cells in your blood. You can kill a lot of virus, bad germs, whatever it is. But today we are trying to make sure like the fusion with macrophage can help the cancer cells. Well, usually macrophage can help you defeat some disease, but this time when they are fused, probably something bad will happen eventually. So in this figure, you can see that after the fusion, there is 1% of cells. Their proliferate rate will higher. They also can obtain resistance to apoptosis. So basically, in the method section, we are trying to do in different sections. First of all, is in vitro experiment section. We do a lot of basic molecular experiments. The first one is we tag the cells with vectors. The vectors can give the cells color, like the red one and green one. So after they fuse, the color will combine as well, and we can see the orange one appear. That is the fusion cell, and then we use flow cytometry to sort the cell. And then we trying to see the fusion of the cell can give the cell some properties. First of all, we do karyotype analysis. We see the chromosome is getting higher, and the flow cytometry, we see the cells stuck in somewhere, can stop them from proliferating. And also we do Western blot RT-PCR to a lot of important proteins and mRNA to see if they can help them to proliferate. And then we do a healing assay and also transwell invasion assay to see if their ability to motivate and move, to see their ability to penetrate some whatever the organism is getting stronger. So next part is in vivo experiment section. We really inoculate the cancer cell in some mice body. But first of all, we see that there's no significant changes compared with the normal cell group. But second, we trying to count and weigh the nodules, any properties about the real stuff. We can see the number and the weight of the nodules in the fused cell group increase significantly. So about the EMT. EMT is actually a really important signaling pathways that can help the cells to develop more, to grow more. Usually when you grow up, the EMT signaling pathways is just there to help you to grow. So we're trying to see the signaling pathways, if the signaling pathways is initiated after the cell fusion. And we do, first of all, we just see it under the microscope. We see the shape is changed, and the transition just happened there. And RT-PCR, we're trying to use RT-PCR to prove that the protein about the signaling pathways is significantly translated more. And also we do further RT-PCR and also Western blot to prove that the EMT related markers, proteins, getting translated higher and higher. So basically, we're gonna have a conclusion. The cell fusion will absolutely enhance malignancy and give the stem cell-like properties. But all the proteins, all the markers related to EMT also have a higher translation rate. Therefore, the EMT process is absolutely initiated. Well, finally, I want to say thank you to my group We published the paper. We made a really good progress. I want to say thank you to my previous supervisor. That's all. Thank you for listening.